Hi guys, Dr. Orlando here with a video on membrane proteins. Should have an S there, huh? And just to give you the layout here, I've drawn this yellow cell membrane, and I've drawn in the phospholipids. I'm not going to draw the cholesterol in here. We know it's there, and I'm not going to draw. I'm not going to draw all of the phospholipids all along the way. We're just going to know that they are there. We're focusing on the proteins here. Now, when I talk about membrane, I'm not just talking about the plasma membrane, the outer membrane of the cell, which is this part. These proteins that I'm talking about, some of them may be on the inner membranes, so on the endoplasmic reticulum, or on a vesicle. Any of those membrane-bound structures that we talked about in class could have any one of these proteins on them. And so keep that in mind. It's not just surface, could be within the cell as well. Okay, so now we're going to look at membrane proteins and look at their jobs. So all of these are jobs membrane proteins could do. All right, so we're going to start with a receptor protein. Okay, so a receptor would look like this. Well, there are lots of different shapes, but this is how my receptor is going to look. It's going to have a very specific shape. Notice that triangle there. Why is it a specific shape? Because its ligand, what binds to it, is specific. So these are one of those lock and key configurations. This is the receptor, and this is its ligand. They fit lock and key style. An example of this might be something like insulin as the ligand, and the receptor would be the insulin receptor. Okay? When insulin binds to its receptor, a shape change would occur in the membrane protein. Remember, proteins, it's all about conformation, so when that binds, the amino acids are tweaked in such a way as to change the shape. And then that will set off a cascade of a reaction inside of the cell. It will signal the cell to do something. In this case, if we're talking about insulin, it's going to signal the cell to take in glucose. And so the cell can use it for energy. So that's a receptor protein. Next up is a recognition protein. Okay, so a recognition protein is, let's erase a little spot here, for ID, so that this cell knows what belongs to it. It can also be used for marking packages for where they should go. So it's either like posted, as we talked about with the Golgi, for example, acting like a post office. So it could be like an address of where this protein should go, or it could be a protein that is identifying self from non-self. Okay, so making sure that we know we've got the right stuff here. So these are often a protein component. So I drew my little square there, but a lot of times they do have a kind of a sugar component hanging off of them, and I'm drawing them branched. And those little green branches would be branches of sugars. Okay, so it's really a protein sugar combination. This is the protein part, and then the sugars are hanging off like the ID tags. Remember, we talked about in class about blood typing and how depending on which sugars are hanging off of the red blood cell tells you whether you are type A, type B, type AB, or type O. And your body knows what you're supposed to be. Your immune system knows what you are supposed to be. So speaking of the immune system, why are these important? Well, imagine a white blood cell like a lymphocyte coming by. And remember I said the immune system are like the police. They go around and they inspect. So it's going to go check these out and ask the question, does it belong here? Okay, does it belong here? It might ask, is this cell making the right stuff? Okay, so it's going to make sure that it's doing the right thing and that it belongs here. And if it doesn't, it's going to take care of it and get rid of it. Okay. All right, those are recognition proteins. Next up, move down the line here, is going to be enzymatic proteins. So enzymes are proteins. They are biological catalysts. So enzymatic proteins. And they are, as I said, biological catalysts, so they speed up reactions. And what's unique about enzymatic proteins, they are an enzyme, and they have a S substrate. It's what they work on. So if this one I'm drawing is going to break down this substrate. So it brings it in, and then the next step, it doesn't move in the plasma membrane as it looks like in my picture here. I'm just showing it as a step here. The next step is this enzyme is going to break down the substrate. Okay, so I broke it in half. Now maybe it makes it so that this can be absorbed more easily. Okay. All right, notice that I made these fit also like a lock and key, because these are enzymes are substrate specific. One enzyme, one substrate. All right, that's the third type of protein. Fourth type of protein, and there are lots of different kinds of these. I'm just going to draw one. Fourth one we're going to talk about is attachment proteins. Okay. With attachment proteins, obviously they are used for attachment. So we're going to draw another plasma membrane here. This is the next door cell. Okay, so we could draw it like that. So it looks like it's going to be attached to this cell right here. And so this guy would have a protein anchor. This guy's got a protein. And then there's also proteins that embed into the membrane, and then they attach to each other like that. Okay, so it's functioning in holding the cells together. This one I drew would be, I know we later on in this chapter we talked about all the different types of attachment proteins. This one I drew would be that spot weld that I talked about, or SNAPs. They're desmosomes. Okay, attaching cells allowing for movement, not watertight. Okay, so that's a protein that's for attachment. And then the last one is the transport proteins. And for transport proteins, I'm going to draw two different ones, and I'm going to need more space. Okay, so for transport proteins, there's two different ones that we're going to talk about. There are lots of types, but these are just two. This one that I'm drawing here is going to be called a carrier transport protein. And we'll make it look like this. Again, this is going to be a specific one. Notice how I give it a very specific shape. With a carrier, the ligand would bind to the protein. That's the ligand. This is the protein. And when it does that, again, this is not moving through the membrane. It's just being drawn next to it as a step. The ligand would bind to the protein, and when it binds, What's so important about one major thing about proteins that I keep saying is that when proteins connect to other substances, they change shape. In this case, it changes shape so that the ligand, the product, whatever it is that they're trying to pick up, is actually released on the other side of the membrane. So that's a carrier. Okay. Binds the ligand, changes shape, spits it out on the other side. All right, last one, then, type of transport protein, or two types of transport proteins that we talked about, carrier and channel. So the channel proteins are just that. They create a channel for something to go through. These channels can be somewhat specific. So for example, if we had a whole bunch of chlorine out here, chlorine ions are negative, and we don't have a lot of chlorine in here, we've set up our concentration gradient, a lot of chlorine on the outside, little bit of chlorine on the inside. If we open that channel, and these channels can be open all the time, or they can be opened with energy or voltage. There's lots of different reasons the channels might open that you'll learn about. We're showing this one just as being open here. Okay, so these chlorine, if that channel is open, will naturally flow down their concentration gradient from an area of high concentration to an area of low concentration. Okay, why do they want to go through that channel? Well, these are proteins. These are proteins, right? So when I draw this box, I want you to imagine that this is really a chain of amino acids that are folded up to make this shape, right? And so imagine all of those amino acids having their R groups in certain places in this blue blob that I've drawn. So what they do is they make sure that they put some positively charged R groups. Maybe a little bigger here. Put some positively charged R groups lining the channel. Right? And if you get positive charges here, those don't look like very good pluses. If you've got positively charged side groups on your amino acids of these proteins lining the channel, those chlorine ions are going to be happy to go through there because opposites attract. All right, so that's what we're going to talk about with transport proteins, the carrier and the channel, and that's all for membrane proteins. So if you have any questions, please feel free to contact me.